Hello, this is a presentation on our vaccine proposal. It's PseudoVax, a novel vaccination strategy, and the Pseudomonas aeruginosa. Pseudomonas aeruginosa is a bacteria, gram-negative, responsible for many cases of different infections. It is a growing threat, especially in the context of antimicrobial resistance. Many proposed vaccines have been put for this microbe, but they did not reach the efficiency goals and they were not put to widespread use. So in this proposal, we describe a novel approach to vaccinating against puriginosa infections, particularly disseminated infections, and also to discuss the efficacy and safety testing.
So Pseudomonas aeruginosa is an important gram-negative pathogen. It has a lot of different disease manifestations. Some of these include burn or wound infections, as well as ventilator or catheter-associated infections in hospitals. The potentially fatal complications from some of these infections can include pneumonia, and as well as disseminated infections that can lead to bacteremia or sepsis. As antimicrobial resistance in Pseudomonas infections becomes more and more common, vaccinations or alternative treatments for infections will become more important. Many virulence factors are associated with Pseudomonas pathogenesis, and it is naturally resistant to some antimicrobials and other aspects of the host's immune system, which means it needs really robust responses for effective clearing of infections. Most important for the previous vaccine attempts have been surface proteins such as OPRF, which is involved in the regulation of virulence, as well as POP-B and its chaperone PCRH, which are major components of the toxin-secreting type 3 secretion system. Flagellin has also been a potential target for vaccines, which is a component of the flagella that allows for motility of Pseudomonas. There have been promising results shown in potential vaccines against this pathogen. However, none of them have been approved or widely used. Many mechanisms of the promising proposals mainly target surface proteins or virulence factors. A previous vaccine targeting POP-B and PCRH, as well as OPRF/I, was able to protect mice against acute pneumonia. And in addition to this, a vaccine targeting OPRI and flagellin proteins provided similar resistance to pneumonia. Vaccines that produce multi-layered responses have historically been the most effective against these types of infections, and mechanisms for stimulating these responses often rely on virulence factor-based vaccines. The figure below shows some of these important virulence factors in Pseudomonas aeruginosa.
For the vaccine, our vaccine name is PseudoVax, and it is a subunit vaccine that will target, of course, aeruginosa infections and for the population at risk. So any people who use like catheters or ventilators, as well as those who are immunocompromised. It aims to produce a Th17 cell response, given the studies have shown that a Th2 response, the normal response against puriginosa, is not effective, and a Th17 response has been working so far. In order to do this, the vaccine components are POP-B, Fly-C, and an adjuvant DMLT. Just to recap, POP-B is a protein found in the type 3 secretion system, which is important for its pathogenesis. Fly-C, as I said before, is a protein for its flagellin. It is also a pathogen-associated molecular pattern. And the adjuvant DMLT is there for its immune-inducing properties. POP-B will be the main antigen target, while Fly-C and adjuvant will be the immune inducer. So, Fly-C is as a PAP, pathogen-associated molecular pattern. It will bind to TLR5 to induce cytokines in the antigen-presenting cell, to induce T cell differentiation into Th17. DMLT also does the same, although to a different manner. It will eventually secrete cytokines for Th17 differentiation. POP-B will be loaded into MHC2 of the antigen-presenting cell, commonly a dendritic cell, to present to it T cell, a naive T cell. When after immunization, dendritic cells will bind to either 5C or the MLT while carrying PUB-B in its MHC class 2. And then the activated dendritic cells will present the antigen to naive T cells in the lymph node, activating them to differentiate into Th17 cells. So when Aeruginosa would infect a vaccinated individual, what would happen is that the antigen presenting cell will load PUB-B into its MHC class 2. And in the lymph tissue, the antigen presenting cell will encounter and bind to memory T cells left over from the previous vaccination. And then the memory T cells would reactivate and then they would proliferate. The reactivated Th cells would then secrete IL-17 and IL-22. These two cytokines, their main function is to clear extracellular bacteria such as P. aeruginosa. And they do this through epithelial production of antimicrobial peptides, neutrophil production, and recruitment to the area of infection, as well as producing even more Th17 cells. In the picture there below, it shows a mechanism of PseudoVax after immunization. And now to the safety and efficacy testing.
To prove the safety and the efficacy of the vaccine, the PseudoVax has gone through numerous intensive safety and efficacy trials. The research team implemented efficacy and safety trials, emphasizing preclinical testing, various phases of clinical trials, and rigorous safety monitoring. To test the safety, animal models such as mice and fish have been selected for the test. These organisms have been previously exposed to the vaccine and then were exposed to the Pseudomonas aeruginosa. And the result of the test was very good. It demonstrated its safety and effectiveness of the vaccine. After the preclinical testing, the 3 different phases of trials were in place. For the first trials, volunteers from the hospital who were already exposed to the infection were separated into a control group and selected group. The selected group were given a small amount of dosage from the research team. However, the control group did not receive any dosage of the vaccine. The health professional staff monitor the health and effect of the vaccine by looking at the recovery speed, white blood cell count, and the overall condition of the patient. The figure below shows that the vaccinated group, which is a selected group from the hospital, all recovered from the infection. However, on the other side, the control group did not receive any doses of vaccine and only partially recovered from the infection. For the next phase, human cells were exposed to the vaccine under the laboratory condition to examine the production of antibodies and activation of immune system components. From this trial, the optimal dosage was found to be at 10 milliliter. For the last phase, the researchers focused on the large-scale evaluation with 32 healthy volunteers from age 18 to 79. This gives the research team variety of the diverse patients and different results. They were examined by the healthcare workers to see the effectiveness of the vaccine by using the same criteria as the first trial. During all these trials and then different phases of trials and preclinical testing, World Health Organization and Safety Committee assessed both the benefits and the potential consequences of administering the vaccines to the public. Thank you for listening to our presentation.